Chapter 14, Section 6, the pentose phosphate pathway of glucose oxidation. Now, the pentose phosphate pathway, or you'll see it in textbooks or old primary literature referred to as the phosphogluconate pathway or the hexose monophosphate pathway, is a pathway that takes our friend glucose 6-phosphate, step 2 of glycolysis, and it ultimately gives us the sugar necessary for nucleotides, DNA, RNA nucleotides. It's a series of oxidative reactions. Remember, this is going to be part of the redox reactions. Reduction is oxidation. So glucose 6-phosphate is oxidized via NADP. NADP is close to NAD that we've seen previously, except this has a phosphate group to it, helps stabilize it, and a few other things biochemically that we're not going to worry about here. So NADP is reduced to NADPH. Glucose 6-phosphate turned into 6-phosphogluconate through a couple of reactions, a few set of reactions. Glucose 6-phosphogluconate is again oxidized, making NADPH, and CO2 is released, so you end up with going from a 6-carbon to a 5-carbon ring. Ribulose 5-phosphate. Ribulose 5-phosphate turned to ribose 5-phosphate, which is, the sugar base for RNA, DNA, and whatnot. We're going to see that ribose 5-phosphate, like I said, RNA, DNA, some coenzymes are going to use this. We find this pathway, this pentose phosphate pathway, used quite often in cells that are carrying out fatty acid synthesis, liver, adipose cells, lactating mammary glands. Okay, so they're going to need lots of NADPH, and this is a great way to generate that very quickly. We also find this, like I said, just liver, but also adrenal glands and gonads for a different reason. They're busy trying to come up with, long hydrocarbon chains to make cholesterol and steroids. This is how they're going to go about doing that. Glucose 6-phosphate dehydrogenase, G6PD. It's going to take, this is one of our first steps in this pentose phosphate pathway, going to take our friend, glucose 6-phosphate. Here they've drawn this looking weird, so they want the straight structure so we can see all the carbon chains. And this weird thing here is to show this is the oxygen bridging between carbon, what is that? Carbon 1, 2, 3, 4, 5. Carbon 5 and carbon 1. NADP, NADPH with our friend G6PD. Again, magnesium there to help balance everything, keep everything happy. NADP reduced to NADPH. And look, we now have a carbonyl at carbon 1. Still joined via this oxygen bridge, or whatever you want to refer to it as. And we have 6-phosphogluco delta lactone. Our 6-phosphogluco delta lactone via the enzyme lactanase is going to be hydrolyzed, so to form 6-phosphogluconate. Our carbonyl, our oxygen here, to our 5th carbon. This is going to be released. Boom. So we now have hydroxyl there. And we have the oxygen up here now. So we have a carboxyl and a hydroxyl. We've gone from 6 phospho 6 glycer, or is it? We've gone from a 6 carbon to a 6 carbon again, but phosphogluconate. So we started off with glucose, and we're now at gluconate. Gluconate is not the end point. Remember, we're trying to get down to a 5 carbon thing. So 6 phosphogluconate dehydrogenase. Okay, we're going to have an oxidation reaction. We're going to pull off some hydrogen, some electrons, reducing NADPH to NADP to NADPH, and we're going to remove a glucose. Which glucose? The one here in blue. Carbon 1 gets released. So now we've shifted it. Carbon 1, 2, 3, 4, 5. What was 2, 3, 4, 5, 6 previously. So we've now gone from glucose 6-phosphate to gluconate, 6-phosphogluconate to ribulose, 6 to a 6 to a 5. And then when it comes down to it, ribulose 5-phosphate, ribose 5-phosphate, basically who's got a hydroxyl, who's got a carbonyl, which is why we have an isomerase and go back and forth, back and forth. So overall, glucose 6-phosphate plus 2-NADP plus some water yields ribulose 5-phosphate, carbon dioxide, because remember we're going from a 6 to a 5 carbon, 2 NADPHs, and some free electrons. Maybe sometimes we need to go backwards. Okay, we got a bunch of ribulose 5-phosphate, but again, we need to get back to glucose. What do? Well, we take the ribulose 5-phosphate and we go through and we start changing things up a bit. What is referred to as an epimerase. Ribulose 5-phosphate epimerase. Can't speak today. I mean, if you look at it, okay, carbon, hydroxyl, carbonyl, carbonyl, carbon, inorganic phosphate, inorganic OH. The only difference is here at carbon 3. Which direction is the hydroxyl in? Remember, shape dictates function. Shape dictates what enzymes can recognize. Ribulose 5-phosphate, xylulose 5-phosphate. Xylulose 5-phosphate, through a series of reactions, can be turned back into glucose 6-phosphate. Xylulose 5-phosphate via trans-ketolase becomes glyceraldehyde 3-phosphate. Trans-aldolase turns it into erythrose 4-phosphate, and then a trans-ketolase with xylulose 5-phosphate, fructose 6-phosphate, which can become glucose 6-phosphate, or fructose 6-phosphate can become. This right here is the part that people get a little lost in. Where are all these things coming? Because if you go through 5, 5, 5, oh, wait a minute, 5, 6, well, even though it's called fructose 6-phosphate, still has 6 carbons. Where are they coming from? Well, you take 2 5s, add them together, you get septidolose 7-phosphate and a glyceraldehyde 3-phosphate. 5 plus 5 is 10, 7 plus 3, 10. Turns out fructose 6-phosphate, which we see right here, fructose 6-phosphate, 6 carbons, it's a 5-carbon ring, but it's 6 carbons, can easily be turned into glucose 6-phosphate. This 4-carbon thing here, boom, recombine it with another xylulose 5-phosphate. 4 plus a 5 gives you 9. Split off 6, fructose 6-phosphate. It's a 5-carbon ring. 6 carbons total. You can turn that easily into glucose 6-phosphate. Whereas the glyceraldehyde 3-phosphate, which you can't see in this figure behind my head, look in the textbook. You take that and a bunch of that and a DHAP, and through the whole gluconeogenesis thing, you get fructose 6-phosphate, you get glucose 6-phosphate. So each of these will add up. 5 plus 5 gives you 10, 7 and 3 gives you 10, 6 and 4 gives you 10. This 4 plus that 5 gives you 9. Here's a 6, there's a 3. And it just keeps adding up. Well, let's look at some of these. Trans-Ketolase. This one's pretty interesting. TPP and trans-ketolase. 1, 2, 3 carbons. And an R group. Here's a carbon there. Trans-Ketolase will then shift those carbons around. R1, R2. Hey, look, R1 now has a carbonyl at its terminal. R2 now has a carbonyl and a carbon with a hydroxyl. So what we just talked about in the last slide, xylulose 5-phosphate plus ribose 5-phosphate. 1, 2, 3, 4, 5 carbons. 1, 2, 3, 4, 5 carbons. Add them all together, yeah, you get 10, but that's not what we want right now, okay? We want something we can work with later. It gives us stuff we can continue to work with. Because if we add them up 10, split in half, we still have our 2 5s. It does us no good. So trans-ketolase is going to transfer these 2 terminal carbons over to ribose. There is remaining 3 here for xylulose. Glyceraldehyde 3-phosphate. We can work with that. The 7, 1, 2, 3, 4, 5, 6, 7, that was ribose is now acetoheptulose 7-phosphate. The 7 acetoheptulose 7-phosphate, the 7 carbon thing here. Well, guess what? Can't do much with that. But if we take that and some excess G3Ps, use a transaldolase to transfer 3 of these carbons, boom, back to glyceraldehyde 3-phosphate. So the 7 and the 3 recombined gives us a 4 and a 6, fructose 6-phosphate. Erythrose 4-phosphate, a 4-carbon, but we can work with that. Our erythrose 4-phosphate from the last slide, we're going to take another xylulose 5-phosphate, recombine these, boom, boom, boom. 1, 2, 3, 4, 5, 1, 2, 3, 4, so 9, shift things around, cut off these 2, transfer them over, because that's what this trans-ketolase, you're transferring a ketol group over, so you're transferring over the 2 carbons. So 1, 2, 3, 4 now becomes 1, 2, 3, 4, 5, 6. What was 1, 2, 3, 4, 5 now becomes 1, 2, 3. More glyceraldehyde 3-phosphate. We can use this. Fructose 6-phosphate. We can turn this back into glucose 6-phosphate. Now, during the previous few slides, you'll notice that TPP would be mentioned, or a protonated shift base was mentioned. What's happening is the electrons that are there, carbanion, is formed. Here you see carbon-carbon double bond, the nitrogen with those electrons, and we see this shifting here. See the same shifting here between the carbon-carbon double bond and the neighboring nitrogen and the lysine. These shifting will help stabilize the intermediates of these reactions. Now, as we've seen before in many of these pathways, these alternate pathways, gluconeogenesis here in the pentose phosphate pathway, there are going to be certain steps that are irreversible. Okay, the energy released during this, the actual reaction itself releases the inorganic phosphate, so you can't get at what's not there. There's a counter to it, a reductive pentose phosphate pathway that converts hexose phosphates to pentose phosphates. See, this one is utilized quite often in the photosynthetic assimilation of carbon dioxide by plants. So that's where this comes into play. Glucose 6-phosphate. Okay. It is the center point in quite a few things going on here. Glycolysis, it's the ultimate goal of gluconeogenesis. It's utilized also by pentose phosphate pathway. And what we see here is that it's the levels of NADPH, which is going to help determine whether or not it goes glycolysis and ATP. High levels of NADPH, well, then this does not need to occur. This can't occur because, well, we've used up all of our available NADPs, because remember, just like with NADs, there's a finite amount there. Thymine is the precursor for the thymine pyrophosphate, the TPP, which makes sense. You know, the TPP have high levels of this. You know, if you don't have high enough levels of thymine, you're not going to have TPP, you're not going to have the pentose phosphate pathway, which means you're going to be struggling to maintain a level of the ribose sugars required for nucleic acid synthesis. We see a couple of human diseases known as beriberi is one of them, swelling, pain, paralysis. Ultimately, it is mortal. Okay, this is a mortal disease. We also see, what is it, Werner? I cannot say it. Characterized by problems with voluntary movements. Okay, heavy drinkers. Okay, heavy drinkers, because it's going to be damage to the epithelial lining of the small intestine, is going to inhibit thymine uptake, which means they're going to have low levels of thymine, and if they have low levels of thymine, they're going to have low levels of TPP, remember, which is one of the cofactors that allows for certain steps that we just talked about in the pentose phosphate pathway, which means lowered nucleotide synthesis and a few other things going on, metabolically speaking. You know, so beriberi, or something that's driven by over alcohol consumption. It's kind of interesting if you know any elderly people who are have been extremely heavy drinkers for decades. You listen to some of their complaints: swelling, pain, you know, it's hard to move, can't feel things, blah blah blah. I'm not saying it's beriberi, but yeah, there's going to be some overlap in their symptoms.